All right. So this brings us to part 7, and part 7 is actually shorter. I just want to give you a taste of how different things work together. And I think I have 3 examples in a row. I want you to sort of review the different things that we've talked about, and this is actually a connection even to a world from dust. Because the very first thing is, what role do metals play in immunity? And actually, let me ask you to make a prediction, because after you've read enough of A World from Dust, these metals will be used in different ways. I'll give you a hint. 2 of these are used to attack, and another one is used to signal. If you haven't read to the second half of the book, you might not know about the one that's used for signaling yet, because it's an advanced function. Immune systems are associated with advanced life, and therefore it is one that's not talked about much in the first half of the book. And maybe that's another clue as well. Do you want to take a guess as to which one of those 3 is the one that you use for signaling, or which one of the other ones is the one that you use to actually attack other cells with reactive oxygen species? Let's see, just give that reactive oxygen species are what 2 of them will do. And it's okay if you get it wrong. I won't tell anyone. You want to guess which one is the signaling one, the one that you haven't seen much, the one that's actually safer for the cell to use? It's a little bit weird because it uses all 3, right? This is one that's used for signaling for advanced organisms that you don't see in the first half of the book.
Zinc.
Yeah, so that's what zinc is. Oh, also it's I should give you another clue. It's the one that you supplement when you have a cold. And again, it's like I'm not quite sure that the supplements work, but in my own experience, I've tried to be very placebo-like with them. And I think that zinc supplements do actually help the immunity. But that's because they help the cells. And my worry about it, how do they get inside the cells? There are importers and stuff like that. But if you're eating the zinc, it still has a ways to go before it gets into the cell where it'll actually help it. But I think it does. So this article is about these 3 metals, and this is it has this picture of innate immunity and adaptive immunity. And just see again, you have macrophages right in the middle, you have B cells and T cells, and you have antibodies being made. We've talked mostly about adaptive immunity, but then my little buddy, the natural killer cell that I studied when I was a postdoc, that is on the side of innate immunity. It's a natural killer, so it's kind of innate, and it's not adaptive. It does not vary its receptor, basically. It's got one and it goes with it. So this is the zinc regulation. I just want you to see. Yeah, there's zinc in the area, and there are zinc transporters, so this must be how it gets into the cell. But most of the zinc is working inside the cell. It's working inside a T cell. It's doing some stuff to metabolism. It's helping, basically. And zinc is used for signaling. When we talk about that a little bit, it's also used for signaling when an egg cell is fertilized. The zinc waves actually go through. There's a bit of that, but it's probably coming up in the reading. The other 2, you see them working, and they're more like they're attacking the cells. They can go inside the cells, but they create reactive oxygen species right here. Iron can activate reactive oxygen species. So this might have some good effects, but a lot of what the iron is doing in immunity is it's acting as a weapon. The same thing for copper. Copper generates reactive oxygen species, less so than iron. Remember that iron is really productive when it comes to ROS. And that's why you have to take care of it. Copper can do it as well. It's redox active. Basically, if it can switch charges. So if you remember your Gen Chem, copper can be copper 1 or copper 2. Iron can be iron 2 or iron 3. If it can switch charges, that means it messes with electrons, and that means it could produce ROS. Manganese is probably the exception to that rule, but it's a pretty good rule and it helps you to remember. You never have to remember zinc 1 or zinc 3, because zinc does not switch its electronic charge very much. Therefore, it's at least redox safe for the cell to use, and the cell uses it like it uses sodium or potassium for signaling. Okay, so I thought that I just wanted to give you a little connection there because I'm making you read that one book, so let's connect it. And the other thing is, we already talked about this a little bit, there's T-cell therapy. The original idea is, why don't we give people T-cells, or more precisely, why don't we select the T-cells that they already make and enrich the good ones and give them back to them, or somehow engineer the T-cells, but basically have a regular T-cell receptor on it. And this is where it started. This is a picture of dividing lymphoma cells, but this is the T-cell engineering is sort of the plain vanilla way of doing it. But then they had an idea that suited them up. They had CAR T-cell therapy. CAR therapy is, the C stands for chimeric. When you see chimeric, that means, do you remember what the chimera was in Greek mythology? Any?
Two different creatures.
Yeah, so a chimeric creature, and the Sphinx technically is a chimeric creature. When you take two different things, and we use that for proteins, if we take a domain from one protein and a domain from another, and we splice them together, we make a chimera. It's not as cool as whatever the chimera actually was, like tail of a snake and stuff like that. But it's tail of a protein, it's that kind of thing. So the C is for chimera, and it's an engineered kind of thing. And you see right here, one of his competitors, Juno Therapeutics of Seattle, Washington, raised $264 million to do CAR T-cell therapy. And my student was involved in them. As the industry goes, he decided to move to another place, and now he's doing something related, but it's also different in many ways. He even uses ViaCore on the job. So he uses FPLC like we use in the lab, and he uses ViaCore. But there have always been problems. T-cell therapy is complex, and a lot of things can go wrong. In 2006, one of the really big things came wrong, and look what happened. It involves cytokine release, which caused organ failure. 2 of 62 patients died as a result of the treatment. And in fact, my student, my alum, who worked at Juno and at other T-cell, he does CAR T-cell therapy now. But he would say that they would use it as sort of a thing of last resort, because they were always aware that if something goes wrong, and they don't always know why, it could result in a cytokine storm. But a lot of these people, for these more experimental ones, they had nothing else to try. So it really was kind of a thing of last resort. The good news is now we know a lot more, and we've figured out how to make it safer. Basically, you take people who are already headed toward hospice, and you give them a chance. And it did actually bring some people back from that stage. So now, this is what a chimeric T-cell is like. So like a regular T-cell therapy is where you're just messing around, but you have basically the same T-cell like this. And you see right there, there's our alpha, beta, and there's associated molecules because it's really, really complex. Of course, it's more complex than I have time to tell you. But on the inside, you have this little tiny cytoplasmic tail that signals to the rest of the cell. This is called the zeta chain. What they do with chimeric T-cell receptors is they just take this part. They take the cross-cellular membrane part, and then they put it onto a different kind of, this is actually an antibody fragment. This is a variable domain of an antibody. So you literally have both of our kinds working together. You have antibodies and antibody on the top, and on the bottom, inside the cell, it's got a T-cell tail. And this will activate the cell, and this will work better because it doesn't have to be restricted to MHC molecules. So now they're selling whole instruments like this, and I just want to say there are things like the antibot. This is not exactly like the antibot. This is something you can buy, and they have whole pages about it where they try to teach you about the technique so that you can do it. I think it's kind of cool that we have, it's an established technology when the companies are selling the instruments to do it. You can look at this if you want to. It's not that different from the antibot, but I don't think that it's going to have a robot. There's too much for the human to do still. We'll see if I'm right about that. And so the very last thing is I just want to remind you that T-cell immunity is inherently complex, There are multiple kinds of TH. Down here you have two different kinds of immunity. And this is TH1 mediated immunity. And this is TH2 mediated immunity. And you see that they're basically dealing with the stuff that we dealt with before. We have antigen presenting cells. We have antibodies being produced up here. We have macrophages. But then we also have mast cells and eosinophils. This is how it all comes together. There's different numbers for each of the interleukins when you look up here. The main thing is, can we do this systematically to try to figure out systematically what different kinds of immune responses we have? Okay. So they did that with this, and they basically took every kind of omics you can think of. So they antibot with proteomics, right? This takes genomics, proteomics, everything, and puts it together, even a methylation array, to try to say, are there different classes of immune subtypes that we can find if we just try to be neutral about it? Not remember the history of immunity, but just take these different omics approach, layer them together, and say, there's really 6 kinds of immune responses. And that's what they did. And they came up with 6 kinds of immune responses. I think it's kind of cool. They basically go together. And this is Institute for Systems Biology. Have you ever seen this? If you're downtown, sort of north of downtown, they used to be in the building that was right across. We could walk there. But this is now, but they moved to be downtown. We've actually toured this place as well. So we've toured Juneau, we've toured. So if you're around next year, we go on a tour, I'll let you know, and you can always hop in. But this is built on the Cancer Genome Atlas. So you see genomics has led to an atlas where they basically, they try to associate genes with outcomes. And they started with 12 different tumor types, you can see them. And they tried to find out which genes were always mutated in that tumor type. And of course it's messy and they overlap and things like that. And they looked at all these different layers. Where were mutations? What were copy numbers? How were they expressed? Where was the DNA methylated? What microRNAs were going on? There's a ton of information, but they did this on a whole cell level, on an omics type level. And the RPPA, by the way, is proteomics. It's a reverse phase protein array, but it's an antibody-based proteomics thing, kind of like the antibody, in the sense of finding lots of proteins that bind. So this was 2013. They had 6 types of omics and 12 tumor types that they did. This more recent one, they expanded it. Do I have 33 cancers? So instead of 13, we have 33. They probably had more layers of data, but they put it all together. And they said, we think we see 6 different kinds of immunity, and this is how they're different. You can have a balanced macrophage to lymphocyte ratio, or you can have a low ratio or a high ratio. You have balanced Th1, Th2 ratio. You can have high amounts of cell proliferation or low. You can have high heterogeneity inside the tumor or low. And then you can have other things that are characteristic of the different responses. But I just think it's fascinating. You can have a wound healing type of response, or you can have an inflammatory type of response. You can have one that's dominated by interferon gamma, or one that's dominated by lymphocyte depletion. Immunologically quiet, I'm not quite sure what that means other than we can't really detect much. Then there's the TGF-beta dominant. These 2 molecules, you'll see a lot of companies going after them. And that's the reason they probably are going after them, because they're 2 major types of immune responses. So all I've got to say is maybe at the end of the day, we can say there's 2 kinds of lymphocytes, then B cells, T cells, natural killer cells. There's 6 kinds of immune responses, and maybe this will be a classification category that we can eventually use for explaining how the immune system works. And the whole thing is, it's built on biochemistry. You need to know your biochemistry and your metabolism to be able to start to interpret this detail, but it's only the beginning. I hope to get you to that point so that then we can go out of here and you can use it in the real world. All right. So that is our structural immunology lecture. Remember that the quiz will be open notes. And the next thing up, if I remember correctly,